Okay, we're back. We're live. It's the 1 o'clock rock. It's Manoa, research in Manoa, and today we have on my far left Toonen. He's an associate researcher in the Hawaiian Institute of Marine Biology in SOWEST, the School of Ocean and Earth Science and Technology. And closer to me is Brian Bowen. He's also with the Hawaiian Institute of Marine Biology in SOWEST. Today we're going to talk about population genetics, evolution, and ecology of marine vertebrates and invertebrates. I get that right? And yep. Brian is the invertebrates man.
I'm the vertebrate man.
Oh, you're the vertebrate man. Okay.
Yeah.
Okay.
Yeah.
I want to be sure. Yeah. And Rob is the invertebrates man.
That's correct.
Okay, remember that and write it down. Okay. So welcome to the show, you guys.
Thanks for having us, Jay.
Great to have you here. We love doing these shows. We learn so much, and hopefully somebody out there learns something too.
Yeah, that's great.
So how did you guys, I need to have people emulate you, study what you do and find out how you got to be where you are so that they can come and get your jobs.
Yeah,
okay.
They can have it.
How can they get your job, Brian? What do they have to do?
Okay. Well, my trail started with a summer home my parents had on Cape Cod. And every day as a kid, I'd go down to the water, and it worried my parents because I'd be in there 4 or 5 hours at a time, looking at all the sea creatures. And Cape Cod's a special place because in the summer, all the tropical stuff comes up from Florida. So you get to see this bizarre marine life that shows up in the middle of summer. And that got me interested in fish. I started keeping fish tanks, and I got an undergraduate degree in biology, then a master's degree. I was intent on studying fish. So I went to College of William and Mary. They have a nice marine lab there. And I got a master's with fish. And while I was getting a master's, there was this inkling of a revolution happening in biology, and that was genetics. All of a sudden, I didn't really care much about DNA, but I figured out you could learn a lot of very cool, important stuff. So I went to University of Georgia, got a PhD in genetics to combine with my fish degree. And then I was a professor and a researcher at University of Florida for 10 years before the offer came here, which I jumped on. Because if you're going to study corals and reefs and fish and stuff, in the United States, it's either Florida or it's here.
Yeah. And you made the choice for here.
Oh, absolutely. Absolutely.
Okay, great. And how about you, Rob?
Well, somewhat similar to Brian, I actually got started with fish tanks, but growing up in northern Alberta, the closest ocean to me was the Arctic. So I did not spend much time there as a child, but my dad owned a pet shop when I was born and used to take me into the pet shop and got me excited about looking after fish tanks. And at some point, about when I was 4 years old, someone said to me, you love your fish tank so much, you're going to grow up and be a marine biologist. And I said, that sounds great. I wonder what that is. And according to my parents, I never changed my mind. I just kept going and really got into it. I did my undergraduate at the University of Alberta in Canada. I moved on to a master's degree at the University of North Carolina, and then a PhD at the University of California in Davis, and was recruited to come here and study population genetics. And like Brian, I jumped on the opportunity to come out here, and I love it in Hawaii. And
yeah, this is the center, isn't it, for your work?
Spectacular place.
So I'm just so curious. You guys are into fish. You're into fish from a young age, both of you. When you go to a restaurant, Alan Wong, for example, or any restaurant that serves seafood, what's your favorite fish?
Without a doubt, my favorite is Ono.
Really?
Oh,
Okay.
that's an awesome fish.
Why? Just the taste? Or are you thinking biologically?
Both. One, this fish is a torpedo. They sit out there offshore, wait for something edible to go by, and when they find it and they take off, their tails move so fast they cavitate. They produce bubbles in the water. So they really are torpedoes. And they're, as the name implies, Ono delicious.
Of course.
So I used to joke, it's not really true anymore, but I used to joke that I'd study fish 5 days a week. Then on the weekend, I'd go fishing.
Of course.
But
the
Ono,
busman's holiday.
Yeah. Ono's my fish.
Yeah, okay. Rob?
Ono is one of my favorites as well. I guess what I eat most often here is local caught ahi. And both myself and my family are big poke fans, and as one of the best managed fisheries that I know of, I think it's responsible and delicious, and I'm a big ahi fan.
In terms of ahi, and I suppose I could ask the same question about Ono, would you prefer ocean caught, in the wild, or would you prefer aquaculture caught?
I can't say I've ever tried an aquacultured.
There are people,
Tuna.
last time I looked, the people who are trying to do
Yeah.
that here. I don't think they've done it yet. But I mean, is there a negative for you between a wild fish and a fish that was grown in aquaculture?
I think it depends on how it's done. So, there are certainly examples. We can think of the shrimp trade, where there are a lot of negatives associated with Southeast Asian shrimp production and the environmental impacts that those
We had Oceanic Institute on the show a few
have.
days
So
ago.
yeah,
Okay, yeah.
so it really depends on how it's done. And for me, that's the bottom line. If I'm going to make a decision, is am I supporting something that I can't agree with or not? And
Yeah.
local line caught ahi, I certainly can agree with.
You can't get any more natural than that.
Yeah, and
Feel
sustainable.
the same way about it?
Yeah, I feel the same way. Nobody's anytime soon going to be culturing an Ono unless you've got a tank that's about 5 miles long, okay?
Because of that cavitation thing.
Yeah, because they are torpedoes. And it's a beautiful, sustainable fishery in large part because they don't school like ahi. They're solitary animals. So you just can't wrap a big net around them and overfish them.
You like their style, don't you?
Oh, I
you
do.
like their sense of independence, their
They're-
hunter-gatherer kind of thing.
They're a classy fish. There was one tagged here on Oahu that was recovered about 1,500 miles south of here, straight over open water.
That's saying something.
This fish travels.
I understand. Okay, so let's talk about your work. Okay, why don't you go first, Brian? What has your work, been focused on? What have you been doing, publishing, and the like?
In the last few years, a big project for us is the origins of the Hawaiian reef fauna, especially for me, the fishes. Now, as you know, we live on a volcano that popped up in the middle of the North Pacific, and that means that everything living here had to come from somewhere else. Every species of organism here is either a colonist or the descendants of colonists. And our question is how they get here. And one of the big theories is that they come over from the West Pacific, Philippines and Japan area. And there's some evidence for that. Rob has a student that graduated that showed our opihi came from Japan a couple of million years ago. And once it got to Hawaii, it radiated into 3 species. Nice story. A lot of the reef fish seem to come from the south, like French Polynesia and the Line Islands. And the most crucial part of that is that about 500 miles south of here is a little dot called Johnston Atoll. And we think it's a gateway into Hawaii for reef organisms, that they hopscotch up from the South Pacific, they get to Johnston Atoll, and from there it's an easy leap to make it into Hawaii. And the
They stay there for a while. Is that they grow strong there and
grow numerous,
grow numerous?
get abundant, get a real strong toehold there on Johnston, put out a lot of larvae into the water, and some of them end up in Hawaii. And that is the gateway into Hawaii. And the reason that matters is because Johnston hasn't always been well treated. It's been
You mean by man?
by man. If you talk to some of the old timers in Honolulu, they'll tell you they used to climb up on their roofs and watch the nukes go off at Johnston. It used to be a place for aerial nuclear tests. Then after that, during the Vietnam War, the US military stored 25,000 barrels of Agent Orange on Johnston. Then after that, Clinton decided he was going to get rid of all the nerve gas. You know, the stuff that by treaty we didn't have?
Oh, that, yeah.
That.
Everybody knows about that.
Right. So
I thought that was just south of Oahu. Margot Edwards, has a whole scientific inquiry going on about nerve gas just south of Oahu.
That's Johnston. And so the island hasn't been well treated. There's some evidence of malformations in the fish there. You'll never see a marine mammal there. It's the only place in the Pacific the marine mammals don't inhabit. And so the gateway to Hawaii, to all this wonderful reef diversity, has not been well treated.
Yeah.
And that's why,
We didn't realize it.
right, and that's why we use the genetics to say, okay, you're a Hawaiian fish. Who's your closest relatives? And a lot of time the answer is Johnston Atoll.
Interesting. Okay, Rob, what's your area?
So
What have you been writing, studying, publishing, whatnot?
most of what my group has been working on lately is trying to understand connectivity in Hawaii. And by connectivity, what we basically mean is if we harvest on Oahu, where's the next generation coming from? Whether that's opihi, whether that's fish. If we eat from our local environment, who's replenishing that with the keiki to grow up to continue to nourish us? And the question is, are they coming from local sources? Are they all on the same island? Are they coming from neighbor islands? Are they coming down from Papahanao-Mokuakea Marine National Monument and moving around? And when I first moved here, Brian and I were hired basically to address some of those questions, to try and figure out, for resource management in Hawaii, where's the next generation coming from? How should we manage our resources? And we use these genetic techniques to say, well, okay, if we look at an individual, if it's a fish or an opihi, where were your parents? Which island were your parents, your grandparents, your great-grandparents on? And if your great-great-grandparents were on Oahu, chances are so will your great-grandchildren. But if your parents are on Oahu and your grandparents are on Kauai and your great-grandparents are up at Kure, then chances are your kids could go anywhere too. And it tells us something about how much individuals move around. Either as a coral, they can't move around much as they're an adult, they're glued down, right? But when they produce their offspring, they're these little microscopic larvae that swash around in the water. And we can't really track them because they're a fraction of a millimeter in diameter and they go somewhere and we don't know where they go. So we want to know if there's a disaster, if something happens, if we have a massive bleaching event or we have a huge coral disease outbreak, or if we eat all of the fish on this reef, where's the next generation coming from? And what most of our research has shown is the idea that those larvae can get everywhere is just fundamentally wrong. And in fact, what we're finding is sort of island by island. In the main Hawaiian Islands, we can't rely on neighbor islands for input of the next generation. If we look at, for example, rice coral on this island, Montipora, the rice coral, common rice coral that we see around, about 95% of the individuals that we test, their parents and their grandparents were on Oahu. Very few of them are coming from neighbor islands. So if we do something, if we have a disease outbreak and we lose the rice coral here, we're not going to get them back for within my lifetime.
Not anytime soon.
And that puts an entirely different perspective on how we go about managing. What happens on Oahu matters on Oahu. We have to look after our own backyard, and what our neighbors on neighbor islands are doing doesn't have as much of an impact about what's going on Oahu, and they can't help us out. If we mistreat our resources here, it's our own fault, and we have to live with the consequences.
That's, you're scientists working with a practical question, a practical problem at the end of the day, so you can recreate our environment if we need to. Regrettably, that could happen.
Right.
Right.
What sort of collaboration do you do together?
A lot. It started off, both Brian and I, so when you say, Brian's the vertebrate guy and I'm the invertebrate guy, it's, for some level, you need to know something about the natural history. So if you want to study an organism, you can't go into something that you know nothing about and try and do a study. And so that's our training and our background is sort of the vertebrates to invertebrates. So Brian does a great job of teaching me all about vertebrates, which I've completely ignored in all of my schooling. And I try and do the same for Brian, and we go back and forth to try to basically work on questions. And over the last 10 years, we've gone through a suite of about 60 common coral reef species across the entire Hawaiian archipelago to try to inform resource management in Hawaii and say, how would it work better if we were to inform the state, if we were to inform federal resource management partners? What should they do? Should they manage the entire thing as one giant archipelago, or should they manage island by island, or should they manage the main and northwest separately? And that's what our research is really focused on, and Brian and I work as partners to try and do that.
That's great. We're going to take a short break. we come back, I'd like you guys to define management for me. How can you, in fact, at the end of the day, we have management here. What is management?
Very good.
Okay, this is Research in Manoa. We have Rob Toonen and Brian Bowen. We're talking about population, genetics, evolution, and ecology of marine vertebrates and invertebrates. We'll be right back.
Aloha, namaskar, and hello. My name is Anu Hittel, and I host the show called Climate Change Beyond Outrage. We go beyond outrage to find solutions to climate problems facing people, nations, and the world. I hope you will join me here every Tuesday at 1 o'clock. We broadcast live from thinktechhawaii.com. Aloha and bye-bye.
Aloha, my name is Carl Kimpania. I am the host of Think Tech Hawaii's Education Movers, Shakers, and Reformers. You can see our show every Wednesday at noon at 12 p.m. on thinktechhawaii.com, as well as visiting YouTube and finding the link for the show there. The show is also aired on OC16. We look forward to seeing you on the show. We have many wonderful guests, including Joan Husted, Corey Rosenlee, where we talk about the very important issues of education for our keiki. We look forward to seeing you there. Mahalo.
Okay, we're back, we're live. We're talking about all kinds of things during the breaks. It's too bad you guys don't get a chance to see what goes on during the breaks. Anyway, so population, genetics, evolution, and ecology with Rob Toonen and Brian Bowen, Bowen at Hawaii Institute of Marine Biology. So yeah, I talked about management, and because I surmise that what you do could have a direct effect on maintenance of our ecology here in case anything goes wrong, and there's a fair chance it will, one thing and another. We should talk about the kinds of things that go wrong. But what about management? What is management? How do you manage? How do I get out there? Do I need a shovel? Do I need a boat? Do I need a net? What do I do?
All of the above. Rob, you want to take the ecosystem management?
So I guess from my perspective, a lot of the history, basically management is about controlling people. What do we do? And
People. It's not controlling fish.
It has nothing to do with nature.
Write that down.
Management is about people, and it has to do with what we're doing. And I think one of the big disconnects from the way that Hawaii was managed prior to Western contact to today was the concept of all of the resources were held in trust by the ali'i, and the management was set down by Konihiki for this is what you're allowed to do. The land, the natural resources belong to everyone, and if you want to take something, you have to ask permission before you do that. I think now our perspective is this is kind of the tragedy of the commons, and basically everyone owns everything, and it's everyone's right to take from the resources without asking anyone else. And sometimes people might have a different view. Maybe some people would like to leave those resources for a future generation, and other people would like to harvest them. And it gets down to the challenge of how do we take what people would like to do with natural resources and find a compromise between what everyone would like out of those resources so that the ecosystem continues to function. Because in the end, if we break the system, it's not there for anyone, and no one gets anything out of it anymore.
God, it's the human condition, isn't it? It's biblical in its own way.
Yeah.
So who is in loco parentis on this? I mean, there was a monarchy once, and the whole structure of the ali'i, I hear you saying that they gave permission for people to use the resources. We don't have that anymore. Who would you say must do it, is doing it, and must do it today?
It has to be the state, but it also, in Hawaii, has to be the federal government. Has to be the one to step up and make the sensible limits and decisions that'll keep that resource healthy for the next 1,000 years. It's got to be that way. And a lot of it now has moved to what they call ecosystem-based management, which is what Rob was talking about. It's getting very close back to the traditional management, where this is your area and you take care of the whole thing. Ecosystem-based management benefits from the genetics because, as Rob explained, if this island gets whacked, will the larvae, the babies, come in from this island and replenish it, or are they isolated ecosystems that have to recover on their own.
Which is harder and takes longer.
Right.
And may not be possible.
Right. That means those ecosystems are relatively fragile. If they get whacked, they don't have that lifeline to other islands. But there's another element of management, which is you can preserve a reef ecosystem for the goatfish and everything else. But what kind of ecosystem can you preserve for a sea turtle? You know, that's migrating 1,000 kilometers back and forth. Or the ono, my favorite fish. There is no ecosystem management for those guys because they move too much. So
Well, the ecosystem is much larger.
oh, the ecosystem is so huge, you can't really do an ecosystem management. That's where you have to have programs designed for those individual animals, like the sea turtles. Mostly, all you got to do is protect that sand where they put their nests. It's not an ecosystem, it's a pile of sand. And once you have that choke point in their life history protected, then those big, highly migratory animals are protected as well.
But when you say protection, again, it comes down to humans.
Yeah.
You want to keep them away from the sea turtle when the sea turtle is laying eggs and raising its young.
Right.
And if you don't do that, then the sea turtle will go extinct.
Right. It doesn't take a PhD in genetics to know that if you're taking all the eggs, pretty soon you ain't going to have the cows,
Yeah.
the adults.
But you know, going back to your point about Johnston Island, if Johnston Island was a relay place,
Right.
then the human management has to be to protect Johnston Island from nuclear, weapons or any other disruptive events. Yeah.
Yeah. Any of those
So I've
environmental
identified
insults.
these relay places.
Right.
And that means, what we talked about before the show, you have to do this phylogeography.
Very good, Jay.
Okay, qu'est-ce que c'est phylogeology?
Phylogeography, Rob?
Geography, yeah.
So philo is basically talking about the genetic lineages, and the geography is maps. So what we're trying to do is map genetic lineages, basically family lines through time. Where did they come from? How did they get to where they are? And where are they found today? So trying to look at those genetic lineages, their distribution in space and time, to understand what are the choke points, what are the valuable places, and if we want to protect Hawaiian reef diversity, where are the places like Johnson Atoll that really matter?
Yeah. I mean, I think one thing that I get out of this is that all of life moves from one place to another. No piece of life on this planet didn't, especially in Hawaii, didn't come from somewhere else. But I mean, human DNA, they can do 23andMe and find out where all the pieces of your DNA came from. And it's the same thing with everything.
Yeah, we're doing 23andMe for the wildlife on the reefs of Hawaii.
Is it as cheap? I think it's $100 for 23andMe.
I'll give you the family rate.
Yeah.
All right.
So how do you do 23andMe for a fish?
What's it cost? About $20 per specimen.
Depending on how we do it.
Yeah.
It's
So you take
somewhere
a fish,
between $20 and $200, depending on exactly how you do it.
and that a fish would be either a vertebrate or an invertebrate. I mean, some
Yeah.
fish are one, some fish are the other.
What we try to do is just take a little tiny tissue clip, let the animal go right where
You're
we
not,
found
you're
it.
not killing the animal to do this.
Sometimes we are,
Okay.
because especially the fish, they won't hold still for you. So really, we can try with nets, and we work really hard with nets, and we've got people that are really good at
To
it.
hold them down.
Just hold them,
Yeah.
take a little clip, that's all. Let them go. But a lot of them,
They don't make it.
it's spearing's the only way we're going to get them.
Yeah.
And
It's easier with the invertebrates when I'm working with corals or sea stars or
They tend
shrimp.
to hold
like
still.
They don't move very fast.
Yeah.
And like a sea star, if you move it off the rock, it's left some tube feet there. That's all we need to get the DNA from it. So with the invertebrates, the non-vertebrates, not fishes, it's a lot easier for a lot of those things we don't even have to necessarily take the animal out of the water.
So you need something under the skin from, deep in the animal, or will the skin or even scales,
Skin.
will that suffice for DNA?
Yeah.
for 23andMe, we could do a swab of your cheek.
Yeah.
That's all we need.
Well, I read recently in the MIT Tech Journal that you can take it off the skin.
Yeah.
Yeah, you
Take
can,
it.
in some cases, you can get it off a glass someone was holding.
Yeah.
Yeah, that's how good it
Amazing
is.
how we've come so far, even in recent years. Yeah.
Yep, and that's all recent technology.
So, okay, so I get the DNA of the animal, vertebrate or invertebrate. What do I do now? What happens?
So basically what we're trying to do now is we're going to extract the DNA from the tissue. So it's exactly what you see on CSI with their,
You mean CSI is true?
CSI is more or less true. It's more expensive and takes longer, and most of the people in the lab aren't quite as pretty. But it's
Right.
basically the same thing. And so what we do is we extract the DNA, we run it through these machines called DNA sequencers, and they give us the DNA sequence.
How do they work?
Well, that's what the breakthrough in technology has been. The way that it used to be, and really in the last 7 years, it's gone from when Brian and I did our PhD, we would work for 5 to 6 years around the clock to get 300 DNA sequences. It was an incredibly arduous process.
What's a sequence? It's the specific genes and chromosomes in
A,
the
C's,
DNA.
G's, and T's, and it's a tiny little piece. What we used to work with was between 300 and 500 letters of a chunk of DNA. So if we think of our genome as the Encyclopedia Britannica, we're picking a sentence at random out of the encyclopedia, and we make 1 billion copies of that so that there's enough of it that we can actually read that sentence. When you're thrown the entire Encyclopedia Britannica, you still find that sentence. That's kind of what it boils down to. And so we look at that chunk of DNA, but that used to take us years of work, because you would run it out on a gel and you would read it by hand with a ruler and a Sharpie and try and get all of the letters and figure it out. And it just was an enormous amount of work. Now, our
Like
students,
using a slide rule.
yeah,
Wow.
Yeah.
and it is,
Wow.
but I
I'm
mean,
glad
now
I
our,
missed that part.
Yeah, me
our
too.
students can generate 1 million sequences in a week.
But by what technology is that?
You basically, now the next generation sequencing, what they do is that they just take, all you need to do is break the DNA into little tiny fragments. And you,
Under a microscope with little tiny instruments.
it's
Electronic kind of.
You just basically, with electronics, you can use sound, you can use a DNA cutter. You just need to make it small. And you can do that as part of the extraction process. And you put it in this machine that bends it into a hairpin and makes a horseshoe out of it. And it reads the sequence, and then it lets go of the horseshoe and it reads it back the other way. And it's in 24 to 48 hours, it spits out millions of sequences.
Calculating the data.
It just
in that
goes
period.
through on a chip, basically. And what it's doing is well, there are different technologies. Some of them are reading the pH, some of them are reading the electrical charge, some of them are reading fluorescence. They do it different ways, but they're
put it all
basically
together.
all just taking and reading the Encyclopedia Britannica. And now the machines give us back the whole Encyclopedia Britannica instead of one sentence.
And this isn't
Yeah.
just in vertebrates and invertebrates research you're doing. This is all across the scientific lot.
It's
Right.
pretty much all come from the Human Genome Project. And
Yes.
all of this pressure to try and get human genomes has spilled down into the rest of the scientific field, and we can take advantage of that now. We're basically we were working in the Stone Age before.
I read only a few days ago that somebody had found a way to modify, I guess, the DNA of a human being to actually cure leukemia.
Yeah.
Quite amazing.
That technology is very preliminary, very experimental.
Right, experiment for sure. It's in Europe, I think.
It
So
has,
it's not here.
yeah, it has the possibility to cure some of these genetic diseases in one generation. Bang, gone. Sickle cell anemia, all these other genetically inherited problems. So the tremendous potential there.
So what they learn about the human genome is relevant to what you guys do. I mean, in terms of the equipment anyway, and the way to sequence, but what you learn is probably relevant to what they do too. No?
Oh, absolutely. What we're doing for these critters, what the biomedical field did for humans. And I'd like to extend that encyclopedia analogy that Rob uses. Every one of these organisms in every one of their cells has an Encyclopedia Britannica that tells us their history, where they've been, what they're doing, why they're so successful. And we just got the key to open that encyclopedia in the last 10 years.
Fantastic. You were at the right time and right place.
Yeah.
Very lucky.
And we are at the right time and right place for a break.
Very good.
Aloha, I'm Kaui Lucas, host of Hawaii is My Mainland, here on Think Tech Hawaii every Friday at 3 p.m. We address issues of importance for those of us who live here on the most isolated landmass on the planet. Please come join me Fridays at 3 p.m. Mahalo.
Aloha, how you doing? I'm Gordo the Tech Czar, here on Think Tech Hawaii, where we co-host Hibachi Talk, where we talk about technology and bring in all kinds of cool guests. Also, my co-host with me today is
Andrew the security guy. Thanks for watching. Thanks for watching Think Tech Hawaii, and thanks for watching Hibachi Talk. We also have Angus.
And you there, lad? It's Angus. I bring in all kinds of wee things. Oh, look, you see my lips moving.
We're back, we're live, we're here with Rob Bowen and Brian, Brian Bowen and Rob Toonen. I mix them all up.
Yeah,
People do that all the time.
we get it a lot.
Good, means you're working well together. All right, so we do the sequencing, we find out what we got in front of us in that animal.
Yeah.
Okay, and what do? You make a printout, you make a computer file? I mean, what do you do with that information?
Well, you build a family tree, except that
Ancestry.com.
yeah, that's all it is. Who begat who? Except in our case, we'll go back 1,000,000 years instead of 1,000 years, and say, where'd you come from? What are you doing that's so successful? But it all starts with field work. And it starts by knowing what's out there, knowing which fish and which corals and which shrimp and clams and all that good stuff.
But it's species by species.
Yeah.
In other words, the project is clams. So we go get some clams.
Right.
We get the DNA of the clams.
Except we'll plan ahead of time to take 30 or 40 species.
Okay.
And then we go to a place like the Papahana Omokukia Marine National Monument.
Way out to the west of Hawaii.
Way out to the
but
west
part
of
of
Hawaii.
the Hawaiian archipelago.
Yeah. We'll go out there on a research expedition, and we're gone for a month at a time. And we get in our dive boats in the morning, and we go out, and we find a reef, look around. Okay, there's a reef, there's one. And we jump in the water. Chances are we're the first human beings to ever see that place.
That must be an interesting experience, a weird feeling.
It's very
It's like
interesting.
You're kind of violating nature, but you're doing it for nature's own benefit.
It's adventure with a cause,
Yeah.
with a good cause.
Yeah.
But there are hazards to that. Our students have been chomped by moray eels. I've had a shark grab one of my flippers.
Well, better your flipper than your foot.
Right. I had a moray eel just about took off one of my students' hands. It was a horrendous situation because you don't know what's out there. You don't know what the currents are. You don't know what the sharks are. And the alua. Up there in the Northwest Hawaiian Islands, you got 150, 200-pound alua. And they look at us and they say, what's that big pink thing doing here?
Yeah.
And they'll come in and check us out and sometimes decide they don't like us.
Yeah. They're big fish.
Oh.
Do they cavitate also?
I haven't seen them cavitate, but what I have seen is one of them decided he didn't like one of the researchers on the expedition. He grabs the guy's dive console, the readout that says how much air he has and how deep he is, grabbed him and just dragged him. Just swam away with one of my scientists. So
That's really unfair.
I did. Yeah.
Didn't he know the good work you were doing?
So that's the start of it, is an adventure. Go out there and get those specimens. And every time, I mean, we've come up, our dive tanks are low on air. We come to the surface and there's no boat there.
Oh,
Things like
to
that.
add insult to injury.
Yeah, not only is there no boat there, but there's black-tipped sharks circling underneath us. Things like that.
And box jellyfish all over the water.
Oh, yeah. I can't count the number of times
Been stung?
they've gotten me.
Oh, my
I think
gosh.
I'm getting immune.
That's entirely possible,
Yeah,
I think.
they sting less now.
Okay, so now you've accumulated all these animals. You take them back, or do you work on them in the field?
Mostly we work on them in the field to collect the specimens and stabilize the DNA. But then, if we're in a situation where we got to kill that animal, we put it in the freezer and we make sure it gets used for science. We bring it back here, people will look at how old it is, what it's eating, how many eggs it produces.
Everything you could learn.
Everything that might help the management.
Yeah, so it's not in vain.
Yeah, we're going to make sure if we have to sacrifice that animal that it's going to get the best conservation value we can get out of it.
Okay, so now you have the DNA.
Now, what do we do?
And you want to make a sort of a comparative analysis or a geographical analysis. What do you do?
Yeah, so, basically you've got each of these things species by species, and it doesn't really help if every time that you look at something it's different. What we see with the opihi is different than with the goatfish and so on. So what we need to do is basically compare all of them and say, well, if we look at all these different species and you're trying to make management decisions, where are the places that are bottlenecks for a large proportion of coral reef habitat? And it might not be every species, but half or 3/4 or 90% of them show a break here. And what we tend to find in Hawaii is basically across the main Hawaiian Islands, the Big Island is different. Maui Nui is different. Oahu is different. Kauai is different. And so
That's
when you
wonderful,
look at
isn't it? It's wonderful to have that kind of diversity.
You know what's
Island by island. I mean, we are so different from the rest of the world. It's a lovely thing, actually.
One of the cool things about what Rob's finding is that channel between the Big Island and Maui Nui, the navigators knew that was a nasty place, that was a tough place to cross. Well, it turns out the fish know it too. Go ahead, Rob.
Yeah. Well, and I think it's one of the things that's sort of changing how a lot of the management agencies are sort of looking at resource management as a result, because the perspective was, well, the islands aren't that far apart. Surely fish can go back and forth there. And for a long time, that has sort of been the perspective, that things must go back and forth between the islands all the time. But more and more what we're finding is actually they don't go back and forth very often. And if we want to manage our resources, it's leaning more and more towards island by island and sort of supporting a lot of these communities who are starting to say, actually, we'd like to set our own regulations. Like, we would like to have different rules for Molokai
than the people.
The people, the communities who live there and who
manages
that area. And they say, we would like to have different rules than Oahu.
Preserve our own species.
That this maybe those are the rules that work on Oahu, but it doesn't work for us here on Molokai. So can you let us have our own rules on Molokai? And that, I think, is something that the research is starting to show a lot of support, that those communities should be allowed to do things differently because what they're doing there matters to them, and it doesn't really impact what's happening on Oahu.
Yeah, that's very valuable going forward because we have to preserve. So, but going back to Johnston Island and going back to the idea that everything in Hawaii is an invasive species or was an invasive species at one point in time, all came from somewhere else. Suppose I find that a given species here in Hawaii has gone extinct because of, we don't have time to go through all the possible ways that could happen, but suppose it happens from one toxic event or another. How do I manage, now that you know on the phylogeographic map where it came from, on the ancestry.com, where it came from, can you go back, say, to, I don't know, Tasmania, I don't know if that's a possibility, and grab some of those animals and bring them here directly, overflying Johnston Island, bring them here and reinstate the population? Can you do that? Is that in the cards? Is this something you guys are looking forward to? Or do you have an idea about how you could, when, where you could do that?
It's called rewilding.
Rewilding.
And yeah, it's starting to happen. There's a group of birds, rails, flightless birds, that were all throughout the Pacific. And when rats were introduced, they were absolutely helpless. They're ground nesting, they can't fly. They were a smorgasbord for the rats, okay? And now that we're getting rat-free areas, we can use that phylogeography to say, who's the closest relative to that Hawaiian rail? And that's the one that should be reintroduced. That's a big, big application of this genetics. Not only just knowing where they came from, but knowing who their closest relatives are if there needs to be transplantation.
And
Rob?
transplantation is complex in the sense that you don't want to go too far off, because then that could disturb the delicate balance of the islands. So you have to have a close relative which fits into the ecology.
Right. And,
Right.
you know, back in the day, the state of Hawaii actually did that experiment when they decided that there were a bunch of snappers and groupers that were missing in Hawaii's nearshore waters naturally, and so they brought in Ta'ape, they brought in Roy, they brought in Ta'au, and they asked, basically, they did that experiment. They brought in quite a few species, and some of them have done incredibly well, and now, talking to most local fishermen, you would say, have had a pretty big impact on some of our nearshore fisheries. Something like Ta'ape is one of the most common fish by biomass in most nearshore waters on our reefs, and it's an introduced snapper that was brought in exactly, as you said, as part of that sort of natural experiment.
Yeah, but part of this, it seems to me, we're almost out of time, but part of this, it seems to me, is to have a snapshot or have a study of what has been here and what has survived and what has gone extinct. Because we want to recreate, the perfect diverse environment as it existed at the top of the throw.
Yeah.
We have to know it was here, and we have to look overseas if we can, and try to bring the missing pieces like the groupers and snappers into Hawaii to recreate the ideal environment for all animals, all ecology.
And I think in this case, there were never snappers and groupers here, and they're actually not part of the natural system, and so they have had a fairly large impact, basically in terms.
But they're not invasive in the sense they're destructive.
I think most people would say they are.
Yeah.
That they're an invasive species and that they're eating a lot. They're a predatory fish that's incredibly abundant and is eating a lot of other fish.
Ooh.
But they're not replacements for anything.
They're not replacements for anything else. They're not causing an extinction of any local snapper. And so, that starts to get into a much more complicated issue in terms of, well, okay, we define invasive basically by what we don't like.
Well, then we'll have to have another show on the subject.
We can go several shows.
Okay, I think you can. I know that. I feel confident. That's Rob Toonen, Associate Researcher, Hawaii Institute of Marine Biology, and Brian Bowen at the Hawaii Institute of Marine Biology on population, genetics, evolution, and ecology of marine invertebrates and vertebrates here in Hawaii. Thank you so much, you guys.
Thanks for
so
having
much
us
for
on the
having
show.
us.
Yeah. Next time we're going to
My
get
pleasure.
a tour of Coconut Island.
Oh, great. We'll do it
Yeah.
there.